be talking about adaptation to salinity today. So we know that salinity is important in limiting distribution and diversification of aquatic organisms. So here we have most groups of gray fin fishes where they're on a log scale and colored by habitat use. So blue is saltwater, red is freshwater. What we see is that most groups are limited to either a fresh or a saltwater environment. That's on a log scale, and if we look at absolute numbers of species within these groups, we see that abundance is dominated by one or two main groups. The largest of these is percomorphs, which make up essentially half of all ray fin fishes. And what we know about percomorphs is that they speciate at twice the rate of other teleosts, and that these high speciation rates are driven by groups in both fresh and salt waters. And so from all of this, I think it's safe to say that salinity is an important factor in generating diversity, and more generally, it's important in aquatic organisms in general, speciation. But what we really don't know is how speciation actually occurs across osmotic environments. And what we can do is look at groups of fish that have managed to make this transition multiple times in order to get an idea of sort of the processes that have generated broader patterns of biodiversity over time. So the group that I'm focused on is Fungilis. And so here we have a phylogeny where branches are colored by osmotic physiology. So blue is marine, green is brackish, and red is freshwater. So what we see is that from a marine ancestor, we have at least 3 independent radiations into freshwater. But what's even more interesting is that we have variation within species for osmotic habitat use as well. So Fungilis heteroclitus is one species that lives in both marine and freshwater environments. And so this is what I'll be focusing on today. So this group is distributed along the entire Atlantic coast of the United States, but I'm focused on populations within the Chesapeake. And so in the Chesapeake Bay, we have a salinity gradient going from the bay, where we have these cool colors signify higher brackish salinities, up into these warmer colors, which are lower freshwater salinities. And within this drainage, we have 2 genetically distinct populations, which I'm calling, I guess because I lack imagination, a brackish water native and a freshwater native population. So these things are genetically distinct. They have a genetic break at a physiologically stressful salinity. There's relatively limited gene flow across this barrier. So throughout my dissertation, and throughout this talk, I'll take kind of the 3 main approaches to disentangle how diversification is occurring within this species. So I'll talk about some physiology, transcriptomics, and very briefly some genomics results. And I hope by taking sort of integrated approach, we can get a really full picture of how diversification is proceeding in a way that any one single approach wouldn't be able to tell us. So first, let's just look at physiology and transcriptomics. So the real question here is sort of how has plasticity changed after selection, sort of in these novel freshwater environments. We might hypothesize that populations should show an increased ability to acclimate to native salinities at the cost of acclimating to foreign salinities. And so to do this, I basically challenged both populations to both low and high salinity stress and looked at their ability to respond. So first, just looking at the low salinity challenge. So on the left here, we have our brackish water native population. On the right, we have the freshwater native population. We have plasma sodium on the top, plasma chloride on the bottom. We use plasma sodium and chloride as they're the 2 main ions involved in that make up your osmolality. Our black lines are our control, and our orange lines are our low salinity challenge. So here, under low salinity stress, where fish are acclimated to high salinity challenge down to freshwater, we'd expect to see a decrease in internal ion concentration. And so for plasma sodium, we see a significant drop in ion concentrations for both our populations. But for chloride, we see only a drop for brackish water native populations and not our freshwater native populations. Looking at our high salinity challenge, where we acclimate individuals to freshwater and challenge high salinity. We see a significant increase for our plasma sodium for freshwater native populations, and no differences for plasma chloride. So looking at these together, we see divergence then for low salinity challenge for plasma chloride, high salinity challenge for plasma sodium. And this suggests that there's some trade-off between the regulation of chloride and sodium homeostasis. And we have some proposed mechanisms that we think might be driving these patterns, and I'd be happy to talk to you guys about what we think those are. So throughout these time course acclimations, we also looked at transcriptomic responses. And so here, I just have a heat map of all of the genes that are showing potentially adaptive responses, so those that differ between populations based on salinity response. So each row is a gene here, each column is a time point. We have our low salinity, our high salinity challenge, broken up by freshwater and brackish water populations. And so within this set of genes, we found a lot of interesting mechanisms that might be driving divergence between our populations, including those involving energetics, which are important for remodeling gills, for cell volume regulation, so dealing with the influx or efflux of water across gills under these different challenges, and also polyamine synthesis, which polyamines are interesting because they're an osmolyte, which buffers ionic changes within your blood, but they're also implicated in driving gill remodeling in response to salinity challenge. So of any of these, this is probably the most exciting one. And again, I'm happy to talk about to anyone about more of the transcriptomics responses if you're interested. So next we can look at sort of broader measures of fitness and more pure physiology, isolated without any transcriptomics. And so we know that osmoregulation is energetically costly, and so we might hypothesize that selection should maximize metabolic efficiency at a population's native salinity. And so to do this, we can look at aerobic scope. And so aerobic scope is an important fitness measure in many organisms. But so if we think sort of about resting metabolic rate throughout time, we know that individuals have small fluctuations, but generally have some minimal resting metabolic rate. Organisms can also raise metabolic rate to some maximum during exercise, and so it's this difference between sort of resting and maximum metabolic rate that's aerobic scope. And this can essentially be thought of as your free energy that you have to put towards things like reproduction, growth, mounting immune response, locomotion, so avoiding predation, capturing prey, and so forth. So basically, to capture this measure, you calculate or you measure resting metabolic rate. You can then chase fish to exhaustion to get maximum metabolic rate, and then just compare the difference between those two values. So first, we can look at the time and the distance that it takes fish to reach exhaustion, sort of as a proxy for their swimming ability. So here we have acclimation salinity on our x-axis, so freshwater and brackish water. Our black lines are brackish water native fish, gray lines are freshwater native fish. And on the left here we have time to exhaustion. And what we see is that brackish water native fish perform significantly better in brackish water than freshwater, where freshwater native fish are not affected. For distance to exhaustion, we see a very similar pattern, but in this case, our brackish water native fish also outperform freshwater native fish at brackish water. So we might expect then that our aerobic scope measure should mirror this performance, right? If you can swim to a certain degree, it would make sense that your metabolic performance would match it. However, that's not the case. Instead, we see poor performance in freshwater, so lower aerobic scope for both populations relative to a brackish water treatment. And so we think that probably the performance differences are being driven by some anaerobic processes that we didn't really capture in this study. And we have some sort of hypotheses about what might be driving this as well. So to conclude with this section, we have found that there's salinity-dependent divergence in swimming ability, which suggests local adaptation. But this is not being driven by metabolic efficiency, at least aerobic metabolic efficiency. And then finally, I just wanted to briefly touch on some genomic work that I'm doing as well. And so I've been talking about this freshwater native and this brackish water native population within the Chesapeake, but what I didn't mention is that there's also a zone of admixture where we have hybrid individuals between the two populations. And so these hybrid individuals, so here we have our freshwater native population here, brackish water native here, and admixed individuals in the middle, where gray refers to freshwater native ancestry, black lines are brackish water native ancestry. So what I'm currently working on is leveraging these admixed individuals to do genotype-phenotype associations. And the idea is that if you have, so admixed individuals are made up of ancestry from both parental populations. They should have large LD blocks, which we can use to hopefully identify adaptive traits in these populations. So this is a work in progress, but it's something I'm really excited about, and I'd really like to talk to anybody about it if you want to know more. So to conclude, we see some divergence in physiological plasticity, where we have a trade-off in sodium chloride regulation. We've identified some transcriptomic mechanisms that we think are underlying those processes. We also see swim performance differences between our populations. It's not explained by metabolic efficiency, but maybe underlying by some sort of anaerobic processes. And finally, just the genetic part of the study that I just mentioned. So to conclude, just thank a lot of people, the Whitehead Lab, and various collaborators at UC Davis and LSU and University of British Columbia, and of course funding sources. If anyone has questions,